Settle in and let your breathing slow, like warm air sinking into a quiet room. You are walking through a greenhouse at night, where the glass holds in the day's last softness. Everything smells faintly of wet soil, leaf dust, and old water sleeping in pipes. Somewhere ahead, a bloom is open, enormous and unhurried, like it has been saving up courage for years. You think flowers are supposed to be sweet and polite and grateful for your attention. Tonight, you learn the calmer truth. This one gets noticed by pretending something has gone terribly wrong. With the sleepless scientist murmuring in the dark, you follow a scent that is not a mistake, but an invitation. And slowly, gently, you will hear how botanists discovered the corpse flower's hidden pollination trick. One patient night at a time. You call it a corpse flower because your language is blunt and your nose is honest. But the plant is not trying to offend you. It is trying to communicate with someone else. And you are just standing in the middle of the conversation, like a person overhearing a private phone call. The first thing you learn as you drift closer is that this smell is not random. It is targeted. It is engineered the way a key is engineered to fit a specific lock. Most flowers spend their short lives bribing bees with sugar and advertising with color. This one chooses a different customer, the kind that arrives when things are complicated. Flies, beetles, insects that make their living off endings. You step nearer, and the bloom looks almost unreal in the dim greenhouse light. It is huge, thick, and folded like fabric gathered into a dramatic curtain. It does not look delicate. It looks like it could survive an argument. And now comes the calmer truth. The one botanists had to learn by watching and waiting and being patient in uncomfortable air. The smell is the headline, yes, but the real story is in the timing and in the way the flower manages its visitors. Because pollination is not romance, pollination is logistics. Your job tonight is to follow the logistics. You keep walking, and the scent deepens, then thins, then deepens again. It is not one smell, but a sequence of notes drifting in and out like a slow song. Meaty, then oniony, then something faintly sweet, as if someone tried to apologize with candy. That shifting matters. Decay is not a single chemical. It is a whole messy orchestra of molecules, arriving on schedule as tissues break down. A dead animal does not smell the same at dawn as it does at midnight. So, the plant does not copy. One moment, it copies the story. Botanists would later measure some of these chemicals and give them long names that sound like paperwork. They found sulfur compounds, the same family that makes onions sharp and eggs suspicious. They found amines, which are common in the smell of rotting fish, and old meat. They found a scatter of other volatiles, small airborne molecules, light enough to travel, and you do not need to memorize the list. You only need to feel the strategy. This flower is painting with smell because it is night and smell travels when color cannot. In the dark, a bright petal is just a rumor, but an odor can move down a corridor, slip under a door, and reach a fly that was not even looking. You think of it as stink. The fly thinks of it as a map, and the map leads here. You can almost imagine the first botanists who took this seriously. Not the tourist with a camera, but the tired plant scientist in a humid greenhouse, in the kind of quiet where every drip sounds like a footstep. They had to start with a simple question. Who shows up when this plant smells like disaster? And the answer was not poetic. The answer was practical. The visitors were scavengers and opportunists and insects that lay eggs in places that do not stay pleasant for long. Because for them, a fresh corpse is not tragic. It is a resource that will be gone soon, so you hurry. That is why the smell works. It creates urgency. You move closer and you notice something else. The air near the bloom feels slightly warmer, not like a heater, not like a sunbeam, more like standing near an animal that is asleep, still making heat without thinking about it. This is not your imagination. Some of these plants can warm themselves. Botanists call it thermogenesis, which is just a fancy way of saying the flower burns fuel to make heat. You are used to plants being cold, unless the sun is on them. But this bloom is spending stored energy to become, briefly, a little warm body. And warmth is not just comfort. Warmth is advertising. Warm air rises and it carries smell with it. It is the same reason you can smell toast from another room when the heat is doing the delivery. So, the bloom heats, the odor lifts, and the message spreads. In the wild, this matters even more because the plant is not in a neat greenhouse. It is in dense forest where air is heavy and night is crowded with damp leaves. If your smell cannot move, your signal dies close to home. So the flower gives the air a push. It is not magic, it is expensive chemistry. You pause and you let your eyes adjust. The bloom is not shaped like the tidy flowers you learned as a child, the kind you draw with a circle and petals. This is an inflorescence, a whole structure pretending to be one flower. The tall central column is called a spadix. It holds many tiny flowers packed close together, like commuters who do not want to make eye contact. Around it is the spathe, the big, dramatic sheath that looks like a petal or a cloak. And the colors are not innocent. Deep red, purple, sometimes a bruised green, shades that look to an animal like flesh or like something rich in nutrients. To you, it is theatrical. To an insect, it is plausible. It is a stage set for a very specific kind of lie. And lies in nature are not moral failures. They are techniques. A fly does not arrive because it wants to admire botany. It arrives because the smell says, food is here, or a nursery is here. Because many flies lay eggs in rotting material where their larvae will have something to eat. If you can trick a fly into believing your flower is a good nursery, it will come quickly. It will land. It will crawl. It will search. And while it does that, it will brush against the real point of the entire performance. Pollen. You stand there and you realize the plant is not begging to be pollinated. It is recruiting. It is hiring temporary workers and paying them in disappointment. The good news for the plant is that the workers do not need to be happy. They only need to move. Botanists had to learn this the slow way. Because these blooms are rare. They do not open every week like a houseplant trying to impress you. They wait. They store energy underground in a swollen stem base called a corm. They grow a single enormous leaf, not a bouquet, but one leaf that looks like a small tree. That leaf photosynthesizes for months, sometimes for years, sending sugar down into storage. Then it dies back, and the plant waits again. And eventually, when it has saved enough, it spends it all at once on a bloom that lasts a short time. So if you are a botanist, you cannot just plan a casual visit. You cannot say, "I will check it next weekend." You have to be ready when it decides. You have to receive a phone call at an annoying hour and drive to a greenhouse in the dark. You have to bring notebooks and patience and the kind of commitment usually reserved for bad relationships. And even then, you might miss the key moment because the trick is not only the smell. The trick is in what the flower does first and what it does later. You lean a little closer and you picture what the botanists were trying to untangle. They had this giant bloom, this dramatic odor, these insect visitors. But they needed more than vibes. They needed a sequence. Which parts are active first? Which parts are active second? When does pollen appear? When is the stigma receptive, the sticky surface that can accept pollen? Because if a flower accepts its own pollen at the same time it releases it, it risks self-pollination. And self-pollination is not always a tragedy, but it can be a dead end. Genetically speaking, it can reduce variation, making future plants less flexible, less resilient. So many plants evolve ways to avoid it, not out of wisdom, out of cold statistical survival. And this is where the corpse flower family, the aroids, becomes quietly clever. Some of them separate the male and female phases in time, not in space, not with walls and fences, with a schedule. You are looking at what seems like one event, one dramatic evening. But the plant is running a two-step plan, stretched across hours. First, it opens in a way that makes the female flowers ready to receive pollen. The stigma becomes receptive, like tiny surfaces turning sticky at just the right moment. But the male flowers are not releasing pollen yet. So if the insect arrives early, it cannot pick up pollen from this same bloom and immediately deposit it back. There is nothing to pick up yet. Instead, the insect might be carrying pollen from a different bloom, from another plant that opened earlier. So the insect stumbles in, lured by the smell, and accidentally delivers pollen to the receptive female flowers. Only later, after that window, the male flowers begin to release pollen. Now the insect gets dusted. Now it becomes a courier for the next bloom. This is called dichogamy, which is a word that sounds like a personality type, but it just means different timing for male and female function. It is a simple idea, but it hides in the chaos of a smelly greenhouse night, and botanists had to prove it by watching the same kind of bloom again and again, and recording the changes. You imagine the notes, time of opening, strength of odor, temperature changes near the spadix, visitor counts, flies and beetles, landing, crawling, leaving, and then the small, careful observations, the kind that feel boring until they become a key. When did the stigma look glossy? When did pollen appear as a dusting? When did the scent peak? Because the plant is not only timing sex, it is timing marketing. The odor tends to be strongest when it needs insects to arrive, and later, when it needs insects to leave, the smell can fade. Not always dramatically, but enough to change behavior. A fly will search longer in a strong odor field because the signal says the resource is close. When the signal weakens, it becomes more willing to give up and move on. So the plant can pull insects in with intensity, then release them with boredom. And boredom, as you know, is a powerful force. You take one more slow step and you notice how the bloom directs attention inward. The spathe curves, forming a chamber, or at least a partial shelter. In some aroids, that chamber can be more than scenery. It can be a gentle trap, not a bear trap, not cruelty. Just architecture that delays, slick inner surfaces, downward pointing hairs, narrow exits, details that make it easy to go in and slightly harder to leave, especially for an insect running on instinct. Because if the insect leaves too soon, it might miss the pollen phase, and then your expensive bloom has wasted its one night of theater. So the plant can hold its visitors just long enough for the schedule to turn, long enough for the male phase to start, and for pollen to become available. You are not watching a flower. You are watching crowd control. You picture the insect experience, because tonight you are not an observer. You are part of the scene. You are a fly guided by odor through dark foliage. Your world is mostly scent and touch, a map made of chemicals. You taste the air with your antennae, and the signal grows stronger. Warmth appears, a gentle gradient, like a hearth you cannot see. You land on a surface that feels soft and ridged. The smell is strongest in a fold, so you crawl toward it. You keep crawling because your instincts say the best part is deeper, and you slip into the central space where the tiny flowers are hidden along the spadix. There is no corpse. There is no meal. There is only the persistent suggestion that you have missed it by minutes. So you search harder. You rub against the floral structures. If you came from another bloom, you may already be carrying pollen, clinging to your body like dust you did not notice. Now you brush that pollen onto the receptive stigma. Congratulations. You have done a job you did not apply for, and you will not be paid. Later, after hours of restless wandering, the plant changes its mode. Pollen becomes available, fine and pale, catching on your legs, your thorax, the small hairs you never think about. And then, when the plant decides it is time, the exit becomes more obvious, or the surfaces change, or the scent simply relaxes. You leave, irritated, and you follow the next promise of decay in the night. And that is how pollen moves between plants that might be far apart. Not by love, by misdirection. This is the kind of story botanists had to build from fragments, because you cannot ask a fly what it experienced. You can only watch behavior. You can set up lights, dim ones, because some insects avoid bright glare. You can record temperature carefully, because the bloom's warmth is subtle but real. You can bag parts of the inflorescence, excluding insects, and see what happens to seed production. You can do hand pollinations, moving pollen with a brush, playing insect with a graduate degree. And over many nights, across many blooms, the pattern becomes a shape you can trust. Scent peaks when the female phase is receptive, drawing in visitors that may carry pollen from elsewhere. Then later, when the male phase releases pollen, the same visitors become dusted. And if the plant can delay them, even briefly, it improves the odds that they stay through the switch. The hidden trick is not one thing. It is coordination, smell, heat, timing, and layout, all cooperating like a quiet machine. You stand in the greenhouse and you let that sink in. The corpse flower is not simply grotesque, it is efficient. And nature loves efficiency, the way a locked door loves a key. One of the calmer misunderstandings people carry is that plants are passive, rooted, helpless. But here you are, watching a plant manipulate animal behavior with chemistry and architecture. It cannot chase a pollinator down the aisle, so it builds an irresistible rumor and lets the insects do the walking. There is a kind of dark tenderness in that. Not moral tenderness, just design. The plant has no brain, no intentions in the human sense. Yet the outcome looks like planning, because selection shapes it as if it were planned. Traits that worked stayed. Traits that did not disappeared quietly, without drama, without an obituary. So the botanist stopped looking for intention and started looking for repeatable steps. And the steps were always small and always physical and always a little rude. You are still standing there in the greenhouse, watching a plant that behaves like a schedule you can smell. You think discovery happens in lightning bolts and champagne and clean diagrams? But here, it happens in damp notebooks and tired eyes and the quiet humiliation of being wrong at midnight. Because the first time you see a corpse flower open, you assume everything important is obvious. Big bloom, bad smell, insects arrive, pollen moves, end of story. Wrong. The important part is the order. And order is hard to notice when you are distracted by rot. So you shift your attention from spectacle to sequence. You imagine the bloom as a theater, and you watch the actors hit their marks. The spathe is the curtain, dramatic and heavy, folding inward like a cloak. The spadix is the tower at center stage, thick, pale, and oddly confident. Down near the base are the tiny flowers, the real machinery, packed tight. And if you want to understand what is happening, you have to stop staring at the costume. You have to watch the hands. You move closer, and your mind starts labeling things, not because labels are fun, but because labels let you keep track. Female flowers first, then male flowers later, in that calm two-step called dichogamy. Stigma receptive, then pollen released. A narrow window, opening and closing like a store that keeps inconvenient hours. And now the question becomes practical, the way good science always does. How did anyone prove this when the bloom is rare and the timing is fussy? How do you catch a plant in the act when the act happens once, then disappears for years? You can almost feel the annoyance of the early botanists. A flower that refuses to cooperate with the calendar. A bloom that opens at night like it is ashamed of daylight. An odor that makes you question your career choices. So you do what people always do with a problem they cannot hurry. You wait. You keep records. You build a small shrine out of data. In the 19th century and early 20th century, a lot of plant science was exactly this. Naturalists with patience, glass lenses, and a hunger for patterns. Later, in modern botanical gardens, it became staff emails at absurd hours. The corpse flower is blooming. Get here now. Bring a thermometer and try not to faint. You show up because you want the truth, and also because the plant will not wait for you. You set up dim lights because bright light changes insect behavior. You position a camera because your eyes will miss things at 3 in the morning. You place a thermometer near the spadix, not touching, just close enough to feel its breath of warmth. You take notes on odor intensity, even though describing smell in words is a cruel joke. Meaty, garlic, old socks in a hot car. And then, beneath the jokes, you add the useful part. When the smell peaks, insects arrive faster. When the smell fades, insects leave sooner. You track visitors the way a bartender tracks trouble. Which species show up? How many? When they land? Where they crawl? How long they stay? Because the flower is not just attracting insects, it is shaping their time inside it. And time is the whole trick. To prove the timing, botanists did something almost tender. They watched the stigma. The stigma is the receptive surface of the female flower, the place pollen must stick. It often looks glossy when it is ready, like a surface lightly coated in syrup. It can even feel sticky if you are careful and slightly disrespectful with your tools. So you check it at intervals and you write down when it changes. Meanwhile, you check the male flowers for pollen release. Pollen is not always dramatic. Sometimes it appears as a fine dusting, pale and dry, like someone knocked flour in the wrong place. If you see pollen too early, your whole neat theory collapses. So you keep checking, and you realize the plant does not care about your theory. It just keeps its own time. There is another method, less poetic, more conclusive. You can exclude insects. You can wrap part of the inflorescence in a breathable bag, fine mesh, tight enough to keep flies out. If no insects can enter, you can see whether seeds form anyway. If seeds do not form, pollinators are not optional. Then you can do hand pollination, using a small brush, transferring pollen on purpose. You become the fly, but with better lighting and worse instincts. You can pollinate during the suspected female phase, then wait. You can pollinate during the suspected male phase and see if anything happens. Over time, the results stop being coincidence and start being a map. Female phase first, male phase later, with just enough separation to discourage self-pollination. And when you compare blooms across years and across plants, the pattern repeats. Not perfectly, because biology is not a factory line, but reliably enough to be called a strategy. Now, while you are imagining these observations, you should know something unpleasant. There is not just one corpse flower in the world. People use the phrase loosely, the way they call many things a weed. There is the famous titan Amorphophallus titanum, enormous and theatrical. There are also other aroids, like some species of arum and others in the same broad family that use similar tricks. They are cousins in the way nightmares have cousins. Some are smaller, some are sneakier, some are more trapped than billboard, but they share a theme. They treat pollinators like delivery drivers, and they optimize the route. When botanists talk about a gentle trap, they are often thinking of those relatives too, because in some of them, the trap is not subtle at all. You are an insect, you follow the smell, and you enter a chamber. And then you discover that leaving is not impossible, but it is inconvenient. The inside can be slick, like waxed plastic. There can be a ring of hairs pointing downward, like a soft fence that works in only one direction. Easy in, harder out. Not because the plant wants to punish you, because it wants to keep you around until the timing is right. You can picture the chamber now from the insect's perspective. It is darker inside and warmer. The odor is concentrated, thick enough to feel like a taste. The walls are close and the surfaces are strangely smooth under your feet. You walk, you climb, you slide, you try again. You are not panicking, not in a human way. You are just repeating behaviors that usually work. And while you repeat them, you rub against the flower parts that matter. You touch the receptive female structures first. Then later, after the plant switches modes, you get dusted with pollen. And only after that, the trap relaxes. In some species, those hairs wilt at the right time, drooping like tired eyelashes. In others, the surface is dry, or the odor changes and you finally give up and leave. Congratulations! You have been detained for pollen processing. Now, to make this feel less like a horror story and more like biology, you zoom in on the mechanics. The plant is not thinking, but it is doing chemistry. Those trap hairs are living tissue, stiff when full of water pressure, softer when that pressure changes. Plant cells use turgor, internal water pressure, like a hydraulic system made of sap. Change the water status, change the stiffness, change the shape. So, a ring of hairs can stand firm for hours, then droop later, simply by changing internal pressure. It is a timed gate made of water and cell walls, and the timing can be controlled by signals inside the plant, hormones, local chemical cues, the slow internal conversation that plants have without nerves. You do not need a brain to run a schedule. You just need parts that respond predictably. Now, the sleepier truth, the one you might not expect, is that the insect is not always miserable. Sometimes it is just confused. Sometimes it finds a little food inside. Some aroids provide small rewards, not nectar like a polite flower, but other tissues or secretions. In some cases, insects may feed on pollen itself. Pollen is protein and fat, a compact packet of plant investment. It is not meant as a gift, but it can be stolen. The plant tolerates that theft as long as enough pollen survives to travel. Because this is the deal nature offers. You can have some if you move some, and the plant stacks the odds by controlling timing and layout. If you are a botanist, this is where you start to see the system as a machine made of wet cells and bad smells, but still a machine with inputs and outputs. Input, stored energy, built up over years. Output, a brief bloom, heat, scent, and a burst of reproductive opportunity. And, in the middle, a temporary workforce of flies and beetles. The machine does not care what you call it. It only cares whether it works. When you read the scientific accounts, the most striking thing is how calm they are. They describe unbearable odor with professional restraint. They quantify temperature rise in the spadix, a few degrees above the surrounding air. They document visitor species, often from families that love decay, like carrion flies. They note when insects arrive and when they leave. They note whether insects carry pollen and where that pollen sticks. Because pollen placement matters. A courier is only useful if the package stays on long enough to be delivered. So the flower structures and the insect's body shape have to match. Hairy legs, textured thorax, small crevices where grains of pollen can lodge like dust in carpet. The plant is not just choosing insects by smell, it is choosing them by physics, by how they crawl, by where they rub, by how much pollen their bodies can carry without noticing. And notice this, because it is one of the quiet revelations, the corpse flower's trick is not a single hidden feature. It is a choreography between plant anatomy and insect behavior. You cannot understand it by staring at the bloom like a sculpture. You have to imagine motion. You have to imagine a fly's feet touching waxy tissue. You have to imagine antennae tasting sulfur compounds in the air. You have to imagine an insect trapped by a simple ring of hairs, waiting out the night. And then you have to imagine the botanist waiting too on the other side of the glass, because the botanist is also trapped, just more politely. They are trapped by the bloom's schedule, trapped by the need to be present at the right hour, trapped by the fact that if they miss this night, they might wait years for the next. So they sit and they watch and they take notes. They do not swat the flies away. They let them crawl, because those movements are the plot. They might even mark some insects, a tiny dot of paint, to see if they leave and return. They might collect a few, gently, to examine their bodies for pollen grains. Under a microscope, pollen is not romantic either. It is geometry. It is texture. It is a surface designed to cling. Each species has its own pollen shape, like a fingerprint, and that lets you answer a crucial question. Is this insect carrying pollen from this plant or from somewhere else? If the insect arrives already dusted, that supports the idea of cross-pollination. If insects leave newly dusted, that shows the bloom is exporting pollen on schedule. This is how the story sharpens. Not from one dramatic bloom, but from many, compared slowly. In some modern studies, botanists also sample the scent itself. They trap the volatiles in small tubes, then analyze them with machines that separate chemicals by mass and behavior. Gas chromatography, mass spectrometry, names that sound like space hardware. But the purpose is simple. Which molecules are present and when do they peak? You might find that certain sulfur compounds surge early, like an opening act. Then others shift later, changing the flavor of the lie. Because a corpse is not one smell, it is a timeline, and the flower is imitating a particular point on that timeline, the point that attracts the right kind of visitor. Some insects prefer fresh decay, a place to lay eggs before competition arrives. Some prefer later decay, where other microbes have already softened the material, so the plant can tune its performance by adjusting the chemical recipe. That tuning is not conscious. It is evolutionary. Generations of plants that smelled slightly more convincing, getting slightly more pollination. Over time, the recipe becomes oddly specific, like a password you did not know you were learning. And in the greenhouse, at night, you stand close enough to be part of the signal. Your clothes pick up the odor molecules. Your hair holds them briefly. You become, temporarily, a moving extension of the bloom's advertisement. If you walk away, you carry that scent down the corridor. You might even confuse a fly, which is both funny and slightly insulting, because it means the trick is working on more than just insects. It is working on air. It is working on physics. It is working on your mammal brain, which is trying to interpret smell as a story, wrought means danger. Rot means disease. Rot means something has failed. And the plant is borrowing that meaning, not to hurt you, but because it is efficient. Now, speaking of efficiency, you turn your attention to something the plant cannot afford to waste: energy. The bloom is expensive, and you can feel that expense in how rare it is. Even in cultivation, even in a botanical garden with perfect watering, the plant does not bloom casually. It builds a corm, a swollen storage organ, heavy with starch. It spends months pushing up a leaf that looks like a small tree, branching, spreading, collecting light. That leaf is the plant's job. It is a solar panel with veins. Every day, it makes sugar and sends it down into storage. And then, often, it collapses and dies back because the plant is not trying to be pretty. It is trying to save. Years can pass like this: leaf, store, rest, repeat. And then, one season, instead of a leaf, the plant sends up the inflorescence. A thick stalk, rising like a slow promise. It grows fast because it is running on savings. It is living off its bank account. And that is why the bloom feels like a single dramatic night. It is the plant cashing out. If pollination fails, it is not just a missed opportunity. It is a financial disaster in plant terms. So, the plant stacks tricks on tricks. Smell that travels, heat that lifts scent and mimics flesh, color and texture that suggests something biologically interesting has happened here, timing that prevents self-pollination and encourages cross-pollination, and sometimes architecture that delays the insect just long enough for the schedule to flip. You can call it grotesque, and you would not be wrong. But it is also simply careful. And careful is what you become if you are a botanist trying to understand it. You start noticing details that casual visitors miss. Not the height of the bloom, but the timing of the odor pulse. Not the drama of the spathe, but the moment the stigma becomes receptive. Not the swarm of flies, but whether they are carrying pollen from another plant. And one of the most uncomfortable details, the one the insects do not advertise, is how willingly they get managed. They are not being forced with violence. They are being guided by their own habits, which is, if you think about it, how most traps work. They do not overpower you. They persuade you to step into the wrong place. Now you let your imagination drift back into the insect body again. You are a fly, and your world is urgency. You do not have long to find a place to lay eggs. You do not have long to find a meal. You follow the smell, because smell is a shortcut through darkness. You arrive at the bloom, and it feels warm, which is a promise. Warmth means metabolism. Warmth means freshness. Warmth means this resource might be new and therefore uncontested. You crawl into the chamber because the odor is strongest there. And inside, the world is tight and crowded with surfaces. You bump into other insects, you climb over them, because politeness is not part of your nervous system. You search for the soft tissue you expect. You search for wet decay, for the right place to deposit eggs. You find none of it, but you keep searching because the signal is so convincing that you assume the problem is you. This is the flower's advantage. A strong signal makes the visitor doubt itself. And while you doubt, you work. You brush against the receptive stigmas. If you carried pollen in, you leave it behind. Later, as the hours pass, you feel something change. The smell is not as urgent. The chamber is still there, still warm, but the story in the air has shifted. And now the male phase begins. Pollen is released, and it clings to you because pollen is designed to cling. You do not know you are loaded with genetic cargo. You only know you are bored and slightly irritated and ready to leave. And that is when leaving becomes possible. Maybe the hairs droop. Maybe you find a gap you missed earlier. Maybe your persistence finally aligns with the plant's schedule. You exit and you fly off into the forest night, following the next rumor. And the plant has exported its package. Now, back in the greenhouse, you think about what it means to call this a gentle trap. Gentle because the insect usually survives. Gentle because the plant is not eating it, not like a carnivorous plant. Gentle because the goal is delay, not death. But still a trap because the insect is not in control of the timeline anymore. It is being held in a biological waiting room. And the waiting room has a purpose: to keep the courier on site until the package is ready. This is where the hidden pollination trick starts to feel less like a curiosity and more like a design principle. You do not just attract the right visitor, you coordinate the visitor's arrival with your internal schedule. You get the delivery at the right time, then you send the courier out at the right time. It is logistics, with smell instead of emails. And because it is logistics, you can actually test it like logistics. You can change one step and watch the whole delivery chain wobble. You can block the smell and see fewer visitors arrive. You can lower the temperature and watch the scent sit heavier and closer. You can prevent insects from entering, and the plant's grand effort stays unanswered. And the strange comfort of botany is that plants will tell you the truth. If you remove their helpers, you picture yourself as the botanist with the mesh bag. You slide it gently over part of the bloom, careful not to tear the tissues. The bag is light, like fabric for a ghost. Air can pass through, but flies cannot. Inside the bag, the female flowers still become receptive on schedule. They still glisten with that sticky readiness, but nothing arrives to touch them. No legs dusted with foreign pollen, no confused beetle doing its clumsy little lap, just empty warmth and wasted perfume. Later, when the male phase begins, pollen still appears. It is released whether anyone is there to collect it or not, because the plant is not waiting for confirmation. It is running a program, and now you see the unfairness of it. The bloom can do everything right and still fail because the world did not cooperate. Nature is not romantic, but it is also not guaranteed. So botanists learn to separate two questions. Can the plant fertilize itself if pollen is present? And can it fertilize itself without an insect to move that pollen? Those are different problems, and plants solve them differently. Some plants are self-compatible, meaning their own pollen can fertilize their own ovules. Some are self-incompatible, meaning the plant has a built-in rejection system, a microscopic bouncer at the door, turning away familiar pollen like it is on a blacklist. In many aeroids, self-fertilization is reduced by timing more than by outright rejection. The plant does not always need a bouncer. It just needs separate shift hours. You let that idea sit for a moment. Separate shift hours for sex. It is not poetic. but it is effective. And it means the botanists had to watch not just the insect traffic, they had to watch the flower's internal clock. They needed to know exactly when female function was active and exactly when male function began. So you imagine the botanist doing something that feels both delicate and absurd, testing stigma receptivity in the middle of the night. Sometimes that meant touching the stigma gently with a tool. Sometimes it meant applying pollen at different times, like tapping on a window. Sometimes it meant later examining whether seeds developed. Because the proof of a receptive stigma is not the shine, the proof is what happens afterward. Whether fertilization actually occurs, whether the plant produces viable seeds, not just hopeful tissue. This is the slow way the story sharpens, not from one observation, but from repeated, boring, careful repetition. A schedule becomes real when it keeps happening. Now you drift back toward the bloom again, and you focus on the visitors. Because the visitors are not just random flies, not if the plant is being selective, and it is. Carrion flies, for example, are drawn to specific chemicals. Some species can detect sulfur compounds at concentrations you would never notice. To you, the greenhouse just smells bad. To them, the air is full of named signals, like street signs. Dimethyl trisulfide, for example, is one of the classic decay cues. It shows up when proteins break down. To a fly, that is not gross, it is information. And a plant that can produce the right mixture becomes a beacon. Not a bright beacon, but a chemical one. You watch a fly land, it does not land like a butterfly, delicate and careful. It lands like someone arriving late to a messy job. It rubs its front legs together, cleaning its mouth parts. It tastes the surface. Flies do that because their feet have taste receptors. Your feet are mostly for stepping on things you regret. A fly's feet are for reading the world. So the insect walks, and each step is a little sample. And you begin to understand why texture matters so much. A smooth surface changes how long the insect stays. A ridged surface guides it. A narrow passage delays it. The plant is not only a billboard, it is a hallway. And hallways are good at shaping behavior without anyone noticing. Botanists notice this when they compare different aeroids. Some species simply lure insects and let them come and go. Others create a clear chamber, a floral room, and inside that room, you find the quiet tools of detention, downward pointing hairs, sometimes called trichomes, wax coatings that reduce grip, angles that make climbing out feel like walking up a greasy slide. If you are a fly, it is not terrifying. It is just inefficient. You keep trying because you always try. And the plant benefits from your stubbornness. Now, the really uncomfortable detail is this. The plant often does not trap just one insect. It traps a small crowd. In the wild, that crowd can include different species, flies, small beetles, sometimes even tiny wasps, all stumbling into the same chamber. And when you pack many insects into a small space, you get friction, not emotional friction, literal rubbing, crawling, bumping, which is exactly what pollination needs. Pollen does not leap gracefully across the air. It transfers through contact, through accidental touch. So the chamber becomes a mixing bowl. a warm, smelly, cramped little room where bodies brush pollen into places it needs to go. And because you are you imagine the human version, a crowded elevator after someone microwaved fish in the office kitchen. Everyone is uncomfortable. Everyone wants out. And because of that, everyone shifts their weight and bumps into each other. Congratulations, you have just modeled pollination dynamics with corporate misery. The botanists, of course, did not write it that way. They wrote about visitation rates and retention times and pollen loads. They collected insects as they exited and checked them under microscopes. How many grains were stuck to the thorax? How many were on the legs? Which body parts made contact with the stigma? Because in many flowers, pollen placement is not random. It is guided by the architecture of the bloom. A fly that crawls inward will touch one region. A beetle that wedges deeper will touch another. And a plant that can steer that contact gets better results. You can almost see the study set up. A quiet greenhouse night, sticky traps placed nearby, not to kill, but to sample. Fine nets to catch insects leaving the bloom. Little vials labeled with time and date. A notebook with times written down in a hand that gets messier after midnight. And in the background, the steady hum of greenhouse ventilation. Not silence, just the mechanical breathing of a building keeping plants alive. This is the setting where the hidden trick becomes something you can describe. You stop saying, "It smells like death, therefore flies come." And you start saying, "The female phase is first," and the scent peak aligns with that. Then the male phase follows, and insects are retained long enough to be dusted. Then the scent declines and the insects leave, carrying pollen away. It is a controlled leak of information. The plant turns the scent on, then down like dimming a light. Now you might wonder how botanists knew the scent changed beyond human noses, because your nose is not the best instrument. It is emotional. It lies to you when you are tired. So they used tools. They sampled the air around the bloom at intervals. They trapped volatile molecules in absorbent materials. Then they brought those samples to instruments that can separate chemicals. Gas chromatography, which is basically a racetrack for molecules. Mass spectrometry, which identifies them by their mass, like checking fingerprints. And what they found was exactly what the fly already knew. The odor is dynamic. Different compounds peak at different times. The bouquet shifts as the bloom moves through its phases. Sometimes the most sulfur-heavy notes come early. Sometimes other compounds rise later, changing the signal from urgent to vague. And that matters, because insects respond differently to different blends. A fly might be pulled by one compound. A beetle might be more interested in another. The plant can tune its guest list without writing invitations. It just changes the air, and the air does the rest. Speaking of air, you finally notice something else about the bloom's warmth. It is not only a comfort detail, it is also a way to build a plume. Warm air rises, and as it rises, it spreads. So the scent does not just sit around the flower like a puddle. It lifts and drifts, following tiny currents through the forest understory. In a greenhouse, those currents are made by fans and vents. In the wild, they are made by temperature gradients and moving leaves and the slow breath of a humid night. A warm spadix is a little engine, not a loud engine, more like a phone left vibrating under a pillow, quietly pushing energy into the air. Thermogenesis costs the plant real resources. It burns stored carbohydrates using cellular respiration just like you do. Sugar plus oxygen becomes energy, plus water plus CO2. Most plants do this at a slow pace because they are not trying to heat themselves. But some aeroids ramp it up. They uncouple the process in ways that release more heat. The energy is not all captured for building tissue. Some of it is spent as warmth on purpose because warmth helps the message travel. That means, for one night, the plant behaves like an animal, not in thought, in metabolism. And botanists, measuring temperature changes, were watching a plant momentarily step into a different role. A quiet imitation of flesh, in more ways than one. Now you pivot back to time, because time is the spine of this whole trick. The plant is not blooming whenever, it is blooming on a schedule it has evolved, and it can be surprisingly consistent. In many aeroids, the first night is the female phase. That is when the stigma is receptive. That is when the odor is strongest. That is when heat production often peaks. The goal is to bring in visitors that already carry pollen from another plant. Then, after that window closes, the plant shifts. The male flowers release pollen later, often the next day or the next night. And by then, the stigma is no longer receptive. So the plant reduces the chance of self-fertilization. Within the same bloom, this is not morality. This is just probability management. You can picture it like two doors in a hallway. One door is open early, ready to accept deliveries. The other door opens later, sending out packages. If both doors were open at the same time, the packages could just cycle in place. Nothing would travel. So, the plant staggers the openings. And suddenly, the smell, the heat, and the trap make sense as one system. Bring the couriers in when you can receive. Keep them close until you can load them. Release them when they are loaded and when they are bored enough to leave. If you are hearing this and thinking, this is too organized for a plant, good. That discomfort is the point. You are still carrying the old fairy tale. Plants as passive, plants as background scenery. But the truth is that plants are chemists. Plants are architects. Plants are patient engineers of behavior. They just do it slowly, without a face, and without your permission. Now, this is where you return to the people who figured it out. Because the plant's schedule is one thing. The human schedule is another. And humans are famously bad at aligning with plants. You imagine the early botanists, before modern cameras, before cheap sensors. They had eyes and clocks and paper. And paper, unlike you, does not get sleepy. It just sits there, holding time still. So they wrote everything down. They described the bloom's appearance at different hours. They noted when the spathe loosened, when it opened wider. They recorded the arrival of insects as the odor rose. They described the smell with the only tools they had, metaphors and grim honesty. Then they returned the next night and checked again. Is the stigma still glossy? Is pollen present? Are insects still trapped? Do the hairs still point inward, or have they softened? This is the kind of work that does not look heroic. It looks like a person standing in a greenhouse at 1 a.m, whispering to themselves and not touching their face. And it required repetition across blooms, because any one bloom could mislead you. Humidity changes odor dispersal. Temperature changes insect activity. A rainy night outside might reduce wild visitors. A greenhouse visitor opening a door might change airflow. So botanists had to compare many events and look for what stayed constant. The constant thing was not the drama. The constant thing was the sequence. And when they finally described that sequence clearly, it stopped being a novelty. It became a mechanism. You are there with them now, in the dim light. You are the person trying to watch insects without disturbing them. You are the person trying to measure a small temperature rise without bumping the bloom. You are the person trying to keep your notes legible as your brain slows down, and you are learning the hard way that biology does not present itself in neat chapters. It presents itself in overlapping signals. The scent rises while the chamber forms. The insects arrive while the stigma becomes receptive. The heat peaks while the odor plume spreads. And you have to untangle it like pulling threads apart with numb fingers. One thing botanists did, quietly, was to separate plant parts experimentally, not in a brutal way, but in a controlled way. For some aroids, researchers could block the chamber entrance, or they could remove or disable the hair barrier, or they could alter airflow around the inflorescence, and then they could compare insect retention. How long do insects stay when the hairs are intact? How long do they stay when the hairs are removed? Does pollen export decrease if insects leave too early? Does seed set drop if insects are not retained long enough to be dusted? You do not need to be dramatic about it. You just watch the numbers shift. And when the numbers shift in the same direction, again and again, you stop calling it coincidence. You start calling it function. The calm power of this is that it turns a weird story into a predictable one. The corpse flower is no longer just a prank on human noses. It is a tuned system designed to solve a hard problem. How do you move pollen between rare plants in dense forests with unreliable visitors when you only bloom occasionally? You cannot afford a sloppy solution, so you evolve a fussy one. And speaking of fussy, you return again to the insects, because the insects are not all the same, and the plant does not attract all of them equally. Some visitors are good pollinators, meaning they actually carry pollen to another bloom. Others are just tourists. They arrive, get confused, maybe feed a little, then leave without delivering anything useful. A good pollinator is one that visits multiple individuals of the same species. Not just one bloom, then a different plant entirely, so botanists had to ask another quiet question. Which visitors are actually doing the job? They checked pollen loads, and they checked behavior. Do these flies travel far? Do these beetles visit more than one bloom? Do they move between plants, or do they just hover around one? Sometimes, they even used fluorescent powders as pollen stand-ins, dusting one bloom, then checking another later under ultraviolet light. It is the kind of experiment that feels like a magic trick, but it is just tracking. Package tracing, again, with better lighting, and the results, in many cases, supported what the plant's design suggested. The main pollinators tend to be carrion-associated insects, insects that already have a habit of moving between smelly resources. That is important because the plant is exploiting an existing route. It is not inventing a new behavior. It is hijacking a routine. And that is what evolution often does. It does not create new desires from scratch. It finds an old desire and attaches a string to it. Now you think about how rare titanum blooms are, and you feel the pressure behind all this. If you bloom once every several years, you are not competing with daisies. You are competing with time. You have one window to export your genes, one window to import genes from another plant, and you are doing it in a world that does not care. A storm could keep insects grounded. A predator could eat your pollinator. A nearby real corpse could outcompete your smell. And that is not even a joke. A plant can lose the scent competition to an actual dead animal. Imagine spending years saving energy, only to be overshadowed by a pig carcass. Congratulations, you have been outperformed by reality. So the plant strategy becomes not just attraction, but reliability. Heat helps the scent travel farther, making it harder to ignore. The chamber helps ensure contact, making visits more efficient. The timing separates phases, making cross-pollination more likely. Each feature slightly increases the probability of success. And when you multiply small probabilities together, you get something that starts to feel like fate. But it is not fate. It is just compounded advantage. Now you drift into the human side again, because the outline of this story is not just plant physiology. It is also discovery, and discovery is never clean. You imagine the first time someone suspected the bloom was running a 2-night schedule, they probably did not announce it with confidence. They probably muttered it. They probably wrote it in the margin. Female first, male later, check, next bloom. And then they had to wait, maybe months, maybe years, to check. That is the particular cruelty of studying rare events. Your hypothesis has to survive boredom. Your idea has to stay alive in your mind long enough to be tested. So you keep your notes, you keep your specimens. You trade letters with other botanists, comparing observations. In botanical gardens, staff share bloom events across institutions because one greenhouse might see a bloom every few years, but many greenhouses together can build a data set across time. And slowly, the story becomes less anecdotal. It becomes comparative. This bloom did this at this hour. That bloom did the same. Visitor counts peaked early. Temperature rose, then fell. In the notebooks, those rises and falls become little waves, repeating across different nights and different plants. You start to notice that the wave is not centered on midnight exactly. It leans toward the hours when the air is cooler and insects are more active. A greenhouse is not a forest, but it still has its own night rhythm. The vents cycle, the humidity settles, the shadows get thicker in the corners, and the flower seems to pick its moment, like it has read the building's schedule. You watch the human side of the operation too, because humans love to complicate a plant's life. A public bloom draws crowds, and crowds change airflow. Doors open, warm, dry hallway air spills in like an insult. The scent plume gets chopped up and carried away. Some botanists started to treat that as an experimental variable, even if they never meant to. On nights with heavy foot traffic, visitor insects behave differently. They arrive slower, or they circle longer, or they leave sooner. Not because the plant changed, but because the arena changed. So, the careful observers begin to do something very unromantic. They control the environment as much as they can. They limit lights. They limit door openings. They keep people at a distance for the sake of both the plant and the data. You stand there feeling the stale warmth on your skin, and you realize how easy it is to miss the real story. A corpse flower is a spectacle, and spectacle invites lazy thinking. Big smell equals pollination. You assume like it is automatic, but the botanists learned to distrust that assumption. They learned to ask if the insects that arrived were actually useful, because not every guest is a courier. Some are just noise with wings. So you picture the next step in the discovery process. You catch the insects as they leave. You do it gently with nets and jars, as if you are borrowing them for a moment. Then you look at them under a microscope, and the microscope has a way of removing poetry from everything. A fly's body becomes a landscape of hairs and plates and tiny joints. Pollen grains cling in the grooves, in the little valleys you never knew existed. And you can count them. Not perfectly, because they are small and stubborn, but enough to compare. You can see which insects leave loaded and which leave clean. You can see which ones actually touched the right parts of the flower. You can even see whether the pollen belongs to this plant species, because pollen has shape. Some grains are spiky, some are smooth, some have little pores, like portholes. A botanist learns to recognize them the way a tired person recognizes familiar keys. And once you can recognize the pollen, you can answer the question that matters. Did this insect bring pollen in from elsewhere? Or did it only pick pollen up here? That difference is the difference between receiving and exporting, between a flower acting as a mailbox or acting as a post office. You begin to appreciate how many ways this could fail. If insects arrive only during the male phase, they leave loaded but deliver nothing here. If insects arrive only during the female phase, they deliver pollen but leave empty-handed. If insects arrive at the right time but leave too soon, they miss the pollen release. So, a flower that can keep them briefly without killing them starts to look less like a gimmick. It starts to look like the simplest way to make rare timing less risky. And this is where the human story, the waiting and note-taking, turns into a kind of quiet obsession. Because once you suspect a trap, you want to see it work. You want to see the insects enter, struggle, and then finally exit at the right hour. You want the schedule to reveal itself like a clock face in dim light. So you do what scientists always do when patience runs thin. You create a version of the world that you can measure. You put a camera on the bloom, point it into the opening. You use infrared light so you can see without changing insect behavior too much. You set it to record for hours, not because you want a cinematic film, because you cannot stare without blinking for an entire night. Later, you watch the footage, and the footage is boring, which is how you know it is honest. A fly walks in, it disappears behind a fold, another fly arrives, then another. Sometimes they leave quickly. Sometimes they do not. Sometimes you see the same insect reappear, climbing, failing, slipping back inside. And when you match that to the timing of pollen release, the pattern gets clearer. Insects that stay through the switch leave with more pollen. Insects that leave early leave with less. The flower is not perfect at holding them, but it does not need perfection. It needs a small improvement in odds, multiplied across visitors. Now you drift back to the plant itself. Because you have been thinking like a person with a clipboard, you stand close again. and the inflorescence looks calm, almost indifferent. It does not twitch. It does not react when an insect bumps it. It simply stays open, warm, and patient, like it is letting the world do what it always does. Inside, though, things are changing. The tiny flowers along the spadix are not all the same. They are arranged in bands, like neighborhoods. In many aroids, the female flowers are lower and the male flowers are above them. Sometimes there are sterile flowers too, structures that do not make seeds or pollen. They just exist to shape the visitor's movement, like furniture in a narrow hallway. That detail matters because it is part of the hidden trick. If an insect walks inward and brushes the lower band first, that is where it delivers pollen. If it later moves upward or wriggles through, it can pick up pollen from the male band. The plant is using anatomy to guide the order of contact, not perfectly, but often enough. And this is where botanists get very particular, in a way that would make a normal person leave the room. They cut cross-sections. They map flower positions. They measure distances in millimeters, which feels absurd next to a bloom the size of a person. But the small distances are what a fly experiences. To an insect, a few millimeters is the difference between touching a stigma or missing it. So the botanist's tiny measurements are actually the visitor's whole world. You imagine one of those dissections done after the bloom has finished its performance. The spade is limp now, the drama gone, the spadix has cooled, the odor has faded into a damp, vegetal sourness. A scientist in gloves takes samples anyway, because the story does not end when the public leaves. They check the flowers for signs of fertilization. They look for swelling, for early fruit development, for the quiet beginning of seeds. If pollination worked, the plant's future becomes visible as a change in tissue. If it did not, the whole structure collapses into compost and the plant returns to waiting. And that is one reason the discovery took so long to sharpen. The consequences of the trick show up slowly. A night of insects becomes a month of fruit growth, or it becomes nothing. So the botanists had to connect fast events to slow outcomes. They had to keep watching after the smell was gone. They had to resist the human urge to declare the story finished when the spectacle ends. You can almost hear the tired satisfaction in their writing when seed set finally matches the timing hypothesis. Not a triumphant shout, more like, yes, that is it. It repeats again. Female first, male later. Retention increases pollen export. Visitor identity matters. And once you have that framework, you can start asking deeper questions, the ones that feel like bedtime secrets. How does the plant know when to heat? How does it coordinate scent production with flower phase? How does it avoid wasting its stored energy on the wrong night? The answers are not mystical. They are made of hormones, enzymes, and the simple reality that plants run on chemistry and light. The plant senses its own size and energy status. It can only bloom when the corm has stored enough carbohydrate. That is not a decision. Exactly. It is a threshold. If you have enough fuel, you can afford the show. If you do not, you keep saving. Inside the tissues, genes turn on and off in patterns that have been shaped by selection. Enzymes ramp up to make scent compounds. Respiration ramps up to make heat. Water pressure changes the stiffness of hairs and surfaces. All of it is coordinated without a nervous system because coordination can be local. Cells respond to nearby signals, and the whole structure behaves like it has a plan. You are tempted to call that intelligence, but it is not thinking. It is architecture plus chemistry, tuned over generations to look like intention. And speaking of things that look like intention, you return to the trap idea because that is where people's minds tend to snag. A trap implies malice, or at least appetite. But in these plants, the trap is usually a delay, not a murder. The insect is inconvenienced, not consumed. That makes the whole arrangement feel almost polite by nature's standards. Still manipulative? Yes, just not terminal. But the details of that delay can be surprisingly physical. Some aeroids use slippery oils on inner surfaces. Some use wax crystals that reduce grip, the way a polished floor humbles your confidence. Some use those downward hairs, which act like a one-way brush. You can push past them going inward, but when you try to climb out, they flex against you, stealing traction. And the really quiet part is how these features can change over time. A hair that is stiff at midnight can be soft by morning. A surface that is slick can dry slightly as humidity shifts. A chamber that feels like a trap can become merely a hallway again when the plant has gotten what it needs. So you are not watching a static structure. You are watching a structure with phases, like the flower itself. The trap is part of the schedule, and that schedule is the hidden trick, the thing that is easy to miss if you only visit during public hours. Because humans love the first night, the big odor night, the headline. But the plant's logistics extend beyond the moment that smells most dramatic. The second phase can be quieter, less theatrical, more about pollen release than attraction. And if you are not there for that, you will misunderstand the system. This is why botanists keep insisting on words like phenology, the timing of biological events. It sounds like paperwork, but it is basically the plant's calendar. And the corpse flower's calendar is inconvenient on purpose. Now your attention slides to a related detail, one that botanists noticed once they started sampling scent carefully. The odor is not just decay, it is warm decay. Not all rotting smells are equal. A cold carcass smells different from a warm one. Warmth changes volatility. More molecules enter the air. Some compounds become prominent only when heated. So by heating itself, the plant is not only pushing scent outward, it is changing the scent's character. It is like warming leftovers and pretending it is a fresh meal. And insects, being insects, respond to those nuances. Some flies are especially drawn to warm, fresh death because that is where eggs have the best chance. Their larvae need soft tissue, microbes, moisture. A warm carcass is a brief opportunity. So, the plant mimics the temperature cue as well as the chemical cue. A lie with props, not just dialogue. That detail helps explain why the bloom can feel almost alive when you stand near it. Not emotionally alive, just metabolically active in a way plants rarely are. You are standing next to a plant that is burning fuel fast, just to be convincing, which is, honestly, a very human way to waste energy. Now you shift, gently, into the uncomfortable detail the insects do not advertise, the one you have been circling. If the flower traps visitors, even briefly, the visitors do not just wait quietly. They stress, not like you do, with existential dread, more like a spike of frantic movement than fatigue, then more movement. And that matters because stressed insects behave differently. They groom more. They scrape their legs. They rub their bodies. They try to clean off whatever is bothering them. And in doing so, they can move pollen around on themselves. They can pack it into hairs. They can smear it onto the right contact points. Or they can lose some. So the trap is not only about keeping an insect present, it is also about increasing contact events. Every attempt to escape is another accidental brush. Every slip is another rub against flower parts. Every frustrated climb is more physical interaction. Pollination as a side effect of annoyance. If you are thinking that sounds like a metaphor for human society, yes, it does. Nature is full of rude parallels. Now you return again to the botanists, because this is where they start to sound less like people describing a weird plant and more like people describing a system. They begin using words like retention time and pollen load and visitation frequency. Those words are dry. They are supposed to be. Dry words are how you keep your mind steady in a room that smells like rot. So you picture a study that tries to quantify retention. You mark insects with tiny dots of paint, different colors for different hours. You let them enter. You watch to see which colors come out and when. You discover that some insects are trapped for several hours. Others escape quickly, depending on their size, their strength, their species. A small fly might slip through hairs that trap a larger beetle. A beetle might bulldoze through a gap that stops a fly. So, the trap is not universal. It is biased. And biased traps are useful because they can select for the best couriers. If a certain insect stays longer and leaves more loaded, it becomes a better pollinator. The plant does not need to consciously select it. The plant's architecture does the selecting automatically. And then, over generations, flowers that retained the best pollinators set more seed. That architecture becomes more common. This is the slow feedback loop that makes the trick feel intentional. No planning, just filtering. Now you let your mind widen a bit, away from a single bloom into the forest where these plants evolved. A tropical understory is not friendly to scent. Humidity can trap odors close to surfaces. Wind is low and unpredictable. Everything is damp and competing. Fungi, bacteria, other flowers, all releasing their own chemical signatures. So if you want to send a message through that crowded air, you need volume, and you need lift, and you need timing. The corpse flower strategy starts to look less like a circus and more like a response to a very hard environment. When you bloom rarely, your margin for error is thin. If you were a plant that bloomed every week, you could afford to be sloppy. Try again next week. Nothing lost. But if you bloom after years of saving, one failure is expensive. It is the botanical version of spending your life savings on a single party hoping the right person shows up. So the plant stacks every advantage it can. It uses scent because scent travels without light. It uses heat because heat moves scent and makes the lie believable. It uses timing because timing reduces self-pollination and increases cross-pollination. It uses architecture because architecture manages visitors without chasing them. And it uses the psychology of insects, which is mostly just instinct plus urgency. Now, without warning, you feel the story shift from discovery to explanation in a softer way, like the lights dimming in a lab. Because once botanists understood the sequence, they could explain the trick as one coherent bedtime secret, not a list of features, but a single coordinated behavior. The plant is running a program. It turns on the advertisement when it can receive pollen. It turns on the heating to push that advertisement outward. It invites the right insects by copying the chemistry of decay. It guides them into a chamber where contact is unavoidable. It delays them just long enough for the male flowers to mature. Then it releases pollen onto them and reduces the incentive to stay. The insects leave, carrying pollen to the next plant, still believing they are following food. That is the hidden trick. Not a secret organ, not a special magical gland, just coordination timed in hours, built out of tissues and molecules. You could explain it in one sentence if you wanted to ruin it, but you do not want to ruin it. You want to sit with the slow elegance of it. A plant that cannot move, moving its genes through the world by borrowing legs. Now, you might be wondering something else, because your brain is annoyingly curious even when you are tired. If this trick is so effective, why do humans still talk about corpse flowers as rare and dramatic? Why are they not everywhere, stinking up every garden? The answer is simple and not comforting. The strategy is expensive, and it only pays off in a narrow niche. You cannot heat yourself and make complex sense for free. You cannot build a massive inflorescence from nothing. The plant needs years of photosynthesis to afford one bloom, and it needs an environment where the right insects exist. Carrion-associated insects are common, yes, but the plant's specific partners may not be everywhere. The scent blend is tuned to certain pollinators, and those pollinators have their own ecology. So the corpse flower is not a universal solution. It is a specialized one, a high-cost strategy for a world where attention is hard to get. And that brings you back to the greenhouse again, because the greenhouse is where humans try to force this strategy into a different context. In cultivation, botanists and horticulturists become part of the pollination story. Sometimes they do not have the right insects inside. Sometimes the insects show up, but not in the right numbers. Sometimes the plant blooms and the public arrives, and the insects are nowhere to be found. So humans intervene. They do hand pollination carefully, like someone trying to repair a tiny machine with a paintbrush. They collect pollen when the male phase begins. They store it briefly, cool, and dry, because pollen is alive and fragile. Then they apply it to another plant's receptive stigma, if they can find one blooming at the right time. And this is where the human schedule becomes its own cruel comedy. Because now you have to synchronize two rare blooms. One plant needs to be in female phase while the other is releasing pollen. If they do not overlap, you have to store pollen and hope it stays viable. You have to keep records and watch corm size and predict bloom timing. You learn very quickly that predicting bloom timing is less science, more weather forecast. You stare at a corm like it owes you a date, and it stays silent. You measure its mass, its firmness, the way it sits in its pot like a buried engine. You watch the leaf phase come and go, like a green flag waving for a season. You wait for the leaf to collapse, because collapse can mean the plant is saving for something else. You check the growth point, the central bud, for swelling that looks different from a leaf start. You become fluent in tiny hints, the botanical version of reading someone's mood from text messages. And still, the plant can decide to surprise you at the worst possible time. It sends up a spear of tissue in the middle of a holiday week. It rises fast, thick, and insistent, like it is spending money it hoarded for years. And suddenly you are not just a scientist. You are logistics again. You coordinate staff. You coordinate temperature controls. You coordinate the least romantic thing in biology, which is pollen storage. Pollen, for all its drama, is fragile. Pollen is alive in a minimal, stubborn way. Each grain is a small capsule holding the male gametophyte, the sperm delivery system in plant form. It is not a seed. It is not a baby plant yet. It is a messenger that can die if you treat it casually. So, you collect it when the male phase begins. You shake or brush it into clean containers because contamination is a quiet killer of good results. You dry it gently because moisture invites microbes, and microbes love free food. You keep it cool because heat makes living cells burn through their limited reserves. You label everything because pollen from different plants is not interchangeable if you care about genetics. And then you hope. You hope the recipient plant enters its female phase while the pollen is still viable. You hope your storage conditions were good enough. You hope the pollen did not clump or mold or silently expire. Congratulations. You have turned a flower into a refrigerated scheduling problem. Now imagine doing this before modern facilities, before dependable refrigerators, before neat plastic vials with screw caps. You are a botanist with glass jars, paper packets, and patience that has been sanded down by time. You try to keep pollen dry with simple desiccants, things that pull water from air. Silica gel is common now, but earlier you used whatever worked, sometimes calcium chloride, sometimes careful airflow. You store samples in cool rooms or underground cellars or ice boxes that sweat and drip. You do it while the greenhouse smells like decay, because the bloom does not care about your comfort. And while you are managing pollen like it is contraband, you are also managing the plant's energy story. Because this is the pivot that matters. This bloom is not a yearly habit. It is a rare expense. So you start thinking about the corpse flower the way the plant thinks about itself. Without thinking, as a storage system, as a budget, as an organism that mostly saves and briefly spends. The corm is the vault. The leaf is the income. The inflorescence is the reckless purchase. And the purchase comes with a deadline. Once the bloom opens, the tissues begin to age. The plant is burning fuel, making heat, making scent, staying upright. That is respiration running hard, like an engine left idling all night. Sugar plus oxygen, making energy, making water, making CO2, and making heat you can feel. And you can feel it because the bloom is large enough to have presence. It is not just warm under a microscope. It is warm in the room, like a quiet animal pretending it is asleep. Botanists who measured this did not do it for poetry. They did it because temperature is a clue to cost. If the spadix heats, it is consuming stored carbohydrate quickly. And if it is consuming stored carbohydrate quickly, that tells you why blooming is rare. A titan arum, for example, can spend years building a corm heavy enough to justify one inflorescence. In the wild, that means years of surviving shade, pests, fungi, and the constant insult of being eaten a little. Because the rainforest does not allow a plant to simply grow in peace. Something always wants a bite. So when the inflorescence finally rises, you are watching a plant cash in survival time. That is why gardens announce it with excitement. Not because it is pretty, but because it is statistically improbable. It is the plant saying, "I have enough saved to gamble." And the gamble is not just about attracting insects. It is also about building an entire structure that can physically function. The spade has to unfurl without tearing. The spadix has to stand without collapsing. The tiny flowers have to mature in the correct order. The tissues have to stay hydrated even while the plant is dumping scent molecules into air. If you have ever watched a cut flower wilt, you already understand the risk. Plants are mostly water, and water is always trying to leave. So the corpse flower has to keep its inflorescence firm through an intense metabolic event. That is part of why it smells like a catastrophe, because it is metabolically close to one. In many plants, flowering is already expensive. In this one, flowering is expensive and theatrical. And theatrical does not mean wasteful. It means specialized. The theater exists because the audience is picky. Carrion insects do not show up for nothing. They show up for certain chemical signatures at certain intensities, in certain conditions. A weak signal is ignored. A confusing signal is ignored. A signal that does not match the thermal cue is suspicious, even to an insect with a brain the size of a crumb. So the plant goes all in. It produces a blend of volatiles that mimic decay, and it does it in a pulse that can reach beyond nearby leaves. It warms the spadix to increase volatility, pushing those molecules into air. And it times it to the hours when insects are likely to fly and forage, because a scent is only useful if someone is awake to read it. Now, step back for a moment, because you are still in the greenhouse. You are watching the bloom, yes, but you are also watching what happens after. The public leaves, the doors close, the air slowly clears, though it never fully forgives, and the plant, once the performance ends, looks oddly tired. The spathe loses its tension. The dramatic folds soften like fabric after a long day. The spadix cools. The smell fades, not into sweetness, but into a dull vegetal funk, like compost that has accepted its fate. If pollination succeeded, the show is over, but the work continues. The female flowers at the base begin to swell. Ovaries enlarge. A fruiting structure develops, slowly, quietly, like a promise made without ceremony. In some aroids, those fruits become clusters of berries, often red or orange, meant to attract birds later. Different delivery system, different couriers. You trick flies at night to move pollen. Later, you bribe birds in daylight to move seeds. Plants are equal opportunity manipulators. If pollination failed, you get the other ending: collapse. The inflorescence breaks down. The plant reclaims what it can, pulling nutrients back into the corm if possible. The rest becomes rot, actual rot this time, not a performance. And then the greenhouse returns to its baseline, which is damp, calm, and ordinary leaves. It can feel like nothing happened, like you dreamed it, which is why the plant's strategy has this Strange, haunted quality. Years of waiting. One night of intensity. Back to waiting. Now you let your mind stay with that waiting, because it explains the whole design. If you only bloom once in a long while, you do not rely on luck. You engineer for bad luck. You assume the forest will not cooperate. You assume insects might be scarce. You assume rain might fall. You assume some idiot animal might find your inflorescence interesting and rip part of it. So you build redundancy into the moment. You increase signal strength with scent. You increase signal travel with heat. You increase contact with structure. You increase cross-pollination with timing. You turn one rare opportunity into a tightly managed event. And botanists, watching this, start to describe it in another language. Life history strategy, resource allocation, reproductive effort, words that sound clinical, but they are describing something intimate. How an organism decides, in its chemistry, when to risk everything. And since you are here for the sleepy version of the truth, you hold on to the simplest picture. Most of the corpse flower's life is a quiet underground bank account. The leaf is the slow paycheck. The bloom is the single extravagant purchase. And the receipt is either seeds or nothing. Now, speaking of nothing, you consider what the plant does between blooms. It does not practice. It does not rehearse. It just waits and builds and tries not to die. The corm sits underground, a swollen stem base filled with starch. That starch is sugar stored in a stable form, like packing energy into dry boxes. The plant uses it later when it needs a sudden construction project. And that is why the inflorescence can grow so fast. It is not growing on current income. It is growing on savings. Botanists have documented Titanarum inflorescences that shoot up startlingly quickly, sometimes inches in a day. It feels unnatural because you expect plants to be slow, but this is the plant spending. When you spend, things happen quickly. When you save, things happen quietly. And if you have ever tried to budget your own energy, you understand the rhythm. Long periods of restraint, and then one day where you do everything at once and regret it. Now, if you are a garden, you feed the plant, you water it, you manage pests, you try to help it save, but you cannot photosynthesize for it. Light is still the income source. So gardens give Titanarum bright conditions, but not harsh sun that burns tissue. They give warm temperatures, because this is a tropical plant that does not want a cold surprise. They give careful watering, because too much water rots the corm, and too little starves the leaf. You can kill it with kindness. You can also kill it with neglect. It is like caring for an introvert who occasionally throws an unforgivable party. And when the leaf is up, it is impressive in a quieter way than the bloom. One giant leaf branching like a miniature tree, catching light across a wide area. It looks like the plant is trying to become a forest of itself, because it is. It needs surface area. It needs chlorophyll. It needs to convert photons into sugar day after day. With no applause, the glamorous part is the bloom. The real work is the leaf. And the leaf, tragically, is what most people ignore. They wait for the corpse, smell like it is the whole identity. But the plant's identity is actually patience. Now you drift into a detailed botanist's love, because it makes the whole thing feel less mystical. Corm size correlates with blooming. Not perfectly, but enough to be useful. A larger corm has more stored carbohydrate and more ability to support an inflorescence. So gardeners weigh corms. They measure circumference. They track growth after each leaf cycle. They keep records like accountants for a silent client. If the corm is gaining mass, you are moving toward a possible bloom. If it is losing mass, the plant is spending more than it earns, and that is dangerous. Sometimes, a plant can produce a leaf cycle that is smaller than the last. That can mean stress. That can mean low light. That can mean the corm is not ready. And sometimes, in a particularly grim twist, a plant attempts to bloom with insufficient reserves. The inflorescence rises, the show begins, and the plant collapses afterward. It spent itself into exhaustion. Congratulations! You have watched a plant burn its savings and face consequences. That is not common in well-managed gardens, but it is part of the biology. Because the plant's internal thresholds are not perfect predictions. They are evolved guesses. In the forest, if you wait too long to bloom, you might die before you reproduce. So sometimes it is better to gamble early, even if you lose. Evolution does not select for comfort. It selects for outcomes across generations. And it is fine with casualties. Now, let your attention slide to the human side again. Because humans do something strange when they meet this plant. They anthropomorphize it. They call it stinky, like it is rude on purpose. They call it obscene, like it has opinions. They treat the bloom as a prank. But if you stay with it long enough, in the dim greenhouse light, it stops being funny. It becomes familiar. It becomes another story of scarcity. Another organism building a strategy around rare chances. And that is where it starts to feel uncomfortably human. You do not always have daily opportunities either. You have a few moments where something matters. And most of life is preparation for those moments. Most of life is gathering resources, learning routines, storing energy. And then, occasionally, you spend it all at once. A test, a job interview, a move to a new city, a conversation you have been avoiding. A plant does not think about these things, but it lives them in its own chemical way. Now, back inside the greenhouse, you remember the smell, and you understand it differently. It is not just gross. It is honest about its function. It is not trying to be beautiful. It is trying to be effective. And effectiveness is often ugly because it is not designed for you. The corpse flower's signal is not meant for mammal comfort. It is meant for insect behavior. It is aimed at a nervous system that equates rot with opportunity. That is why the scent is specific. That is why it is timed. That is why it is paired with heat. And that is why it can feel like the plant is whispering to the dark, not to you, through you. Now, there is another layer, softer, and somehow worse. The corpse flower's performance depends on a whole ecosystem of decay. The insects it recruits are not random scavengers. They are part of nutrient cycling. They are part of the process that turns dead bodies into soil. They are the cleanup crew that keeps ecosystems from choking on their own remains. So when the plant mimics decay, it is piggybacking on an ancient pipeline. Death to larvae, larvae to flies, flies to pollination, pollination to seeds, seeds to new plants. It is all one loop, nothing is wasted except maybe dignity. And if you are the kind of person listening to this in bed, you probably know the loop is not comforting, but it is calm in the way machinery is calm. It runs whether you watch it or not. Now you watch the greenhouse return to normal, and the normal is almost boring. Leaves sit still, pots hold their soil, water lines hum softly. The bloom, if it is done, sags like a deflated myth, and the corm underneath, unseen, begins the long work of recovery. It will send up a leaf again. It will rebuild reserves. It will wait. It will become, once more, a quiet plant with no smell, no crowd, no drama, which makes the whole thing feel like a secret. A plant that lives most of its life as a boring green budget sheet and then briefly becomes a scandal. And the botanists who studied it, the ones who waited through nights, understand that boredom is the real baseline. The bloom is an interruption. The leaf is the lifestyle. Now you drift to an uncomfortable comparison that botanists rarely say out loud, but they think it. This strategy is like advertising during a crisis. You do not put up billboards every day. You put up a billboard when you need attention fast, and because attention is expensive, you make the billboard unforgettable. The corpse flower is unforgettable, not because it is kind, because it is efficient at hijacking the right instincts. And that is why when you zoom out, it is not a freak. It is a specialist. It is a plant that solved a problem with the tools available: chemistry, heat, timing, structure, patience. Now, with all that in mind, you let the scene become small again. You are standing close enough that you can see texture in the spathe, tiny variations in surface, like skin that was never meant to be touched. The light is dim, and the red looks darker, like dried blood in shadow. The air is thick with humidity, and the remaining scent clings to the back of your throat. You think about how many years of sunlight went into this, how many days of quiet photosynthesis, one leaf collecting light like a solar sail, how many nights the corm sat underground, doing nothing dramatic, and then how quickly it all gets spent. That imbalance is the heart of it. Years for days. Savings for spectacle. Now, without any warning, your mind wanders to another small human detail. The people who work in botanical gardens learn to read the plant's mood, not because it has emotions, but because it has patterns. They learn the look of an inflorescence bud versus a leaf bud. They learn that an inflorescence bud is thicker, more blunt, more like a fist than a spear. They learn that the plant's growth rate can hint at what it is building. They learn to set up barriers before the public arrives because the smell will attract humans the way rot attracts flies. Different species, same weakness for spectacle. And they learn to plan for the aftermath because if the plant sets seed, it will need support. It may need staking. It may need careful nutrition. Fruit development is another cost. More spending, more time before the next leaf, more delay before the next bloom, which means every successful pollination can also push the next spectacle further away. Success has a price. It always does. Now, hold on to that thought because it leads you into the softer part of the story, the part where this odd flower feels human in the dark. Not because it is noble, not because it is wise, because it is trying. It is trying in the only way it can, with a body built for stillness. It cannot walk to find a mate. It cannot chase a pollinator. It cannot call out in sound. So it speaks in molecules. It speaks in heat. It speaks in timing. It speaks in inconvenience. And if you listen to that, if you really listen, you can hear something familiar. A desire to be carried forward, not emotionally, biologically, to persist, to not end here, in this pot, in this patch of forest, in this one body. And you realize that the grotesque theater is just packaging. Underneath is the simplest biological urge there is. Make another one. Now, here is the sardonic twist. Nature does not care how you do it. Nature does not reward beauty. Nature rewards function, and it will accept almost anything that works. If bees are reliable, you make nectar. If birds are reliable, you make berries. If flies are reliable, you make rot. If you need to hold them for a while, you build a room and make the door inconvenient. It is not elegant in the moral sense. It is elegant in the mechanical sense. And botanists do not solve this like a puzzle with a final click. They listen long enough to hear the repeating beat. They watch enough blooms to separate accident from design. They measure enough odors to stop trusting their own noses. They wait through enough nights to see the schedule repeat. And when they finally describe the hidden trick, it sounds boring on paper. Timed female phase, timed male phase, thermogenesis, volatile emission, insect retention.